Hello guys, today we are going to see nucleic acid from biomolecule chapter. What are nucleic acids? Nucleic acids are unbranched polymers of repeating monomers called nucleotides. In other words, nucleic acids have a polynucleotide structure. Nucleotide contains three things, that is base, sugar and phosphoric acid. Name the two types of nucleic acid. Where are they found in living cells? So the two types are RNA and DNA. RNA full form is ribonucleic acid. DNA is deoxyribonucleic acid. RNA is mainly found in the fluid of living cells, that is cytoplasm, while DNA is found primarily in the nuclei of living cells. Which sugars are present in RNA and DNA? So I have already told you that nucleotide contains three things: sugar, base and phosphoric acid. In RNA, sugar present is D-ribose sugar, while in DNA, sugar present is deoxyribose sugar. So these are the structures of ribose and deoxyribose sugars. Just you can replace this OH by H. Other things are same only in both the sugars. Ribose is present in RNA and deoxyribose is present in DNA. Next is nitrogen bases present in nucleic acids. So I just discussed now that sugar, base and phosphoric acid. So bases present in nucleic acid are purines and pyrimidines. Purine base is adenine and guanine. Pyrimidine base is cytosine, uracil and thymine. Uracil only occurs in RNA while thymine only occurs in DNA. Other three bases are common in both RNA as well as DNA. And then structures of these bases we are going to study. First is diagram for a pyrimidine base. So these are three pyrimidine bases. See the first one, cytosine, then uracil and then thymine. So how you can draw pyrimidine bases? So these two nitrogens, okay, and in between them there is a carbonyl group, C double bond O. Nitrogen always makes three bonds. In cytosine, NH2 is present. In uracil, C double bond O is present, while in thymine it is similar to uracil, only this CS3 group is added. Okay, thymine is present in DNA, uracil in RNA, and cytosine is present in both. Similarly, draw a neat diagram for the purine bases in nucleic acids. Okay, so these are the purine bases, adenine and guanine. In adenine, 4 nitrogens are in the ring and NH2 group is there. In guanine, same 4 nitrogens are there. Apart from that, C double bond O is there instead of NH2, and NH2 is placed between these two nitrogens in guanine. So that's how we can draw the structures of purine and pyrimidine bases which are present in DNA and RNA. Pyrimidine bases are cytosine, uracil, thymine. Purines are adenine and guanine. Next is nucleoside. So nucleoside contains only two things, base and sugar. And nucleotide, we already discussed, it contains phosphoric acid also. A nucleoside is formed by joining the anomeric carbon of the sugar with nitrogen of base. So we can clearly see the first carbon of sugar is anomeric carbon. Anomeric carbon is also called as chiral carbon. 4 different groups are attached to this carbon, therefore it is anomeric. And cytosine is the base used here. So see the attachment point, 1 and 1 dash. OH of ribose and H of cytosine goes in the form of water and a ribonucleoside is obtained. So nucleoside basically contains sugar and base. In pyrimidine bases, the nitrogen atom at one position bonds with the 1 dash carbon of the sugar. Then other examples are 2-deoxyribose plus adenine gives deoxyribonucleoside. Again same thing happens, anomeric carbons OH and adenines hydrogen from this nitrogen. So water goes out and deoxyribonucleoside is obtained. In purine bases, the nitrogen atom at the 9 position bonds with 1 dash carbon of the sugar. Now next part is nucleotides. Nucleotides contain base, sugar and phosphate group, that is generally phosphoric acid only. Nucleotides are formed by adding a phosphate group to the 5-OH of a nucleoside. Thus nucleotides are monophosphates of nucleosides. So AMP is adenosine monophosphate, base, sugar and phosphoric acid. Okay, so this we already discussed in nucleosides. Only thing that CH2OH from this part. So hydrogen from that and OH from the other. So water again goes out. So adenosine monophosphate, because only one phosphoric acid group is attached. And in this example, deoxycytidine monophosphate. So deoxyribose is there, cytosine is there and monophosphate is there. Here adenosine, then this is ribose, so therefore it is not mentioned, and monophosphate is there. Likewise we have to name these nucleotides. And now we are going to discuss structure of nucleic acid. Nucleic acid, both DNA and RNA, are polymers of nucleotides formed by joining the 3-OH group of one nucleotide with 5-phosphate of another nucleotide. The ends of polynucleotide chain are distinct from each other. One end having free phosphate group of 5' position is called 5' end, and the other end is 3' end and has free OH group at 3' position. The primary structure of nucleic acids is the sequence of the nucleotides in it. This in turn is determined by the identity of the bases in the nucleotides. Different nucleotides have distinct primary structure. It is the sequence of bases in DNA which carries the genetic information of the organism. The polynucleotide chains of nucleic acid are named by the sequence of the bases beginning at 5' end and using the 1-letter symbols of the bases. For example, the name CATG means that there are 4 nucleotides in the segment containing the bases cytosine, adenine, thymine and guanine in the indicated order from 5' end. So that's how these are the few points from structure of nucleic acid. So we'll stop here. Thanks for watching the video.